Good morning, everybody. My name is Rahul Rajala, and today I will be talking to you about some molecular biology parameters and their differences and relationships. The three parameters I will be discussing today will be rate, processivity, and fidelity, specifically when it comes to DNA polymerase. Now, as you all know, DNA polymerase adds nucleotides to a DNA strand. So first, I think it'd be easier if I drew it out for you. Let's draw a replication fork, okay? So now we have our two DNA strands, and we have them being broken apart right here. Then we have our 5' here, 3' here, 3' here, 5' here. We have a 5' here, then a 3', and then we have here we got some 3', 5', 3', 5'. So what we have here is this is the leading strand, and that's the lagging strand. And as you can see, the lagging strand is composed of Okazaki fragments, which will eventually be ligated together. And the leading strand will not have Okazaki fragments, since it's being read from the 5' to 3' end, and DNA polymerase works from the 5' end to 3' end. Alrighty then. Now, first, let's talk about rate. What does rate mean? Rate refers to the number of nucleotides per second. Thus, the number of nucleotides incorporated into these strands in a given time frame. So it's almost like a velocity term, you could say that. And the rate varies. For a human eukaryotic cell, the DNA rate might be somewhere in the region of 50 nucleotides per second. In humans. In bacteria, however, they have been shown to go as high and maybe even higher, 1,000 nucleotides a second, a whopping 20 times faster than humans. Processivity, on the other hand, refers to it's a characteristic of enzymes that function on polymeric substrates. In the case of DNA polymerase, the degree of processivity refers to the number of nucleotides added each time the enzyme binds to a template, or the number of nucleotides added per catalytic event. Thus, think about it like this. It's as if the DNA polymerase binds and then runs the length for a little while and then drops off, and then it binds again and then runs the length for a little while and drops off. And this happens over and over again. Processivity refers to that particular length. So a higher processivity means the DNA polymerase can run faster. Once it's bound, a non-processive DNA polymerase adds nucleotides at the rate of 1 nucleotide per second. Processive DNA polymerases, however, can add multiple nucleotides per second, drastically increasing the rate of DNA synthesis, and the degree of processivity is directly proportional to the rate of DNA synthesis. So this is just nucleotides per catalytic event. And for Pol1, we have a processivity of maybe 10 to 100 nucleotides per second. And for Pol3, we have maybe about 1,000 nucleotides per second. My mistake, drop the seconds. Just 10 to 100 nucleotides and 1,000 nucleotides. And you might ask, why does Pol3 have such a better, processivity? Well, Pol3 is a hollow enzyme in which the primary enzyme is involved in DNA replication in E. coli and belongs to the family of C polymerases. And it consists of 3 assemblies. The beta sliding clamp, which increases the processivity, there we go right there, and the Pol3 core, and the processivity factor, and the clamp loading complex. The core consists of 3 subunits, the alpha, epsilon, and the 5 which acts as a stabilizer for the epsilon. The holoenzyme contains 2 cores, one for each strand, lagging and leading. Thus, basically, because Pol 3 has a beta clamp, we can represent it like this. It'll attach itself to the DNA and basically string along the DNA, allowing for greater processivity. And this can also be seen at the replication fork. There is an increase facilitated by DNA polymerase association with proteins like the beta sliding clamp. Now, last but not least, let us talk about fidelity. Fidelity refers to the number of incorrectly placed nucleotides for the number of correct. Now, these will be usually 1 out of 100, so in that case equal to like 10 to the minus 2. So we could say the fidelity of 10 to the minus 2 is bad and 10 to the minus 3 is better. So, and fidelity is initially based in the active site geometry. So if we have like a DNA, we have an enzyme, and then the DNA polymerase enzyme, and then we have, the DNA through, and then we have a base. If we remember our Fisher induced fit model, it will induce a fit with this empty base right here, and it'll try to, match that. The enzyme will try to match that. However, this is not necessarily the greatest way to go about it, but it does work. And because of this, we have a fidelity of about 10 to the minus 4. So 1 out of every 10,000 is incorrectly placed. However, this is not great. I mean, it seems good, 1 out of 10,000, that's a great deal. But when you think about it, DNA is heritable material. If changes occur, changes will stay and be passed on to daughter generations. And over time, these mutations can lead to perhaps a terminatable organism, an organism that just doesn't have the facility to survive because of a buildup of these mutations. So, evolutionarily speaking, this is bad. This is why there is a 3 to 5 exonuclease domain on DNA polymerases, on some DNA polymerases, and this increases the facility of the DNA polymerase's fidelity. Thus, it can basically cause a 100-fold to 1,000-fold increase in fidelity because it's a proofreading enzyme. It'll proofread the template afterwards, and this changes our fidelity from 1 to the 10 to the minus 6 to 1 to the 10 to the minus 7. Jesus, that's amazing right there. That's 1 out of, 10 to the minus 10 to the 6. That's 1 out of a million. And 10 to the minus 7, 1 out of 10 million. And we all know that there's 3 billion base pairs. So that's still, a sizable amount of errors, but nothing that's, too horrible. And I think that's about all for all of this. I hope you enjoyed this mini lecture, and I bid you all a safe and pleasant evening.